Um, for being here. I'm Sasha Reed. As Peter said, I'm incoming to the Wild Department and Director of the Center for Colorado River Studies. And it is my absolute pleasure to introduce Dr. Noah Fierer. Noah is a true scientific rock star in every sense of the word. So he's a professor at the University of Colorado Boulder. He's also director for the Center for Microbial Exploration. He has crushed every metric we use to assess the impact of scientists on the field. He's written hundreds of papers, received millions and millions of dollars in grants. He's won many awards. He just won the ISME Winogradsky Award, which is a big deal. But none of that really speaks enough to the effects that he's had on the field of microbial ecology. He has transformed our understanding of what microbes are where, what they're doing, how they're responding to and causing change in soil, water, atmosphere, buildings, the human body. He's absolutely an incredible mind in the field. My favorite thing about Noah is how fun and adventuresome he is. He's a fantastic collaborator, mentor, dad, skier, mountain biker. And I think that his sense of adventure is part of what makes him so important as a researcher in the field. He's willing to let ideas fall down and get back up again. He's willing to think outside of the box. So I'm so delighted that he's the first speaker for the Ecology Center's 2026 events. And please help me and welcome Noah.
Thanks, Sasha, for the introduction. It was a little cringy sitting over there, as my daughter would say, but no, thank you. And thank you for the invite, and thanks for all the graduate students that are organizing this and the invitation to come. It's been great meeting with a lot of you, and I look forward to chatting with more of you later this afternoon and tomorrow. Should be fun. So we're really thankful. So I just want to sort of expand on what Sasha talked about, and that my group, I'll say my group, works on microbes in a lot of different systems. And generally, if I had to summarize what we do, it's exploring microbial diversity, protists, bacteria, fungi, and linking those microbes to ecosystem, plant, and human health. And we work in a wide range of systems. I get bored easily, so it's nice to work in lots of different systems, from like tap water to microbes found in our homes to microbes found in the near surface atmosphere, outdoors, looking at permafrost thaw and sort of how microbes are responding to permafrost thaw. We've also done a lot of work on like plant microbe interactions, like how the plant microbiome shapes the chemistry of plants and the flavor of harvested crops, and like the seed microbiome and so forth. So we work in lots of different systems, but today I want to talk about my favorite system, my native habitat, soil. Okay. So I recognize that a lot of you probably don't think that much about soil. I can't blame you. And maybe you don't think that much about microbes, and you most likely don't think a lot about soil microbes, right? But I want to pose a question for you. Okay. And it's okay if you don't know the answer, because I've posed the same question to people that make their living studying soil bacteria. And the answers are all across the board. So you don't have to answer it yet, but I'm going to ask you to place your best guess. And this is a question: what is the average generation time of bacteria in soil? Okay, this is a fundamental ecological question. If you study plants and animals, most likely you have a good understanding of how fast those plants and animals grow and reproduce, right? And understanding generation time is super critical for understanding how organisms, regardless of the organism, whether it's a microbe or a plant or an animal, interacts with the environment and interacts with other organisms. Right? So this is the question I want you to think about for the next few minutes. What is the average generation time of bacteria in soil? Because again, this is a fundamental topic in ecology and just in basic natural history, right? Okay, but first some background. But think about this. I'm going to have you place your bets in a minute. So just some background. So first of all, when we're talking about generation time in bacteria, we're talking about doubling time. How long it takes for a single bacterial cell to reproduce and become two cells. Bacteria, like many microbes, can also grow. They can expand their cell size, but that's not what we're talking about here. We're talking about generation, so new bacteria forming. Okay, so if we look at bacteria that we study in the lab and can grow in the lab, for example, on a Petri plate, we see about 2 orders of magnitude variation in generation times. So on the one side, we have E. coli. It's useful as a model organism because it grows really fast, about 15 to 20 minute generation time, 50 to 20 minute doubling time. And this is why we study it so much in the lab, because it grows. You can have vats of this in just a day or two. On the other side of the spectrum, we have Treponema pallidum. Anyone know what this bacteria is famous for? Causes syphilis. Okay. This bacteria is incredibly difficult to study in the lab. It will grow in the lab, but it has like a 1.5 day generation time. So to study it in the lab and to get enough biomass to do experiments and so forth, it could take weeks or months. So it's really frustrating to work with. So across bacteria that we routinely cultivate and study in the lab, this is what we're working with. Okay, this is sort of the range in generation times. The other thing I want to point out, I pose this question, what is the average generation time of bacteria in soil? And the key word there is average. So in any given community, plants, animals, bacteria, fungi, there's obviously going to be some range in generation times. There's going to be organisms that have very short generation time and organisms that have very long generation time. I don't know what that distribution looks like. Let's just assume it's a normal distribution. So what I'm asking you is what's the average? Okay. Then now is when you get to place your bets. Okay, so generation time on the x-axis, less than a day to more than 10 days. How many people think the average generation time of bacteria in soil is less than 1 day? So for reference, we have E. coli over here, and the syphilis bacteria, which has about a 1.5 day generation time under ideal conditions here. Less than 1 day, raise your hand. Okay, 1 to 5 days. Few, 5 to 10. How about more than 10 days? 1 lone vote in the back. Okay. I'm not going to reveal the answer. Well, first I'll preface this by saying we don't really know the answer to this question. So it's kind of like, my apologies. But I think we're getting a better idea of what this answer might be. And I want to first start by presenting some sort of anecdotal evidence that we think it's actually pretty long. And one thing I want to try to emphasize is that most soil bacteria are likely oligotrophs. What's oligotroph? What does that mean? Okay, so how do we define what are these oligotrophic soil bacteria? So one definition is organisms that are evolutionarily adapted to exploit ecological niches characterized by low substrate concentrations and low energy flows. So organisms that grow slowly. They grow efficiently, but slowly, and they're adapted to these low resource conditions. So not like E. coli. Another definition, E. coli, which is a classic copiotroph, which is the opposite of oligotroph, sort of fast growing, built for maximum potential growth rate, has evolved a feast and famine existence. Oligotrophic bacteria are never invited to a feast. So even when resources may be available, they can't grow fast. Again, they're built for efficient growth, not rapid growth. Okay. And so what I want to highlight here is that there's this metabolic trade-off. So like all traits, there's trade-offs, right? There's this trade-off between optimization for growth rate, which we would see like copiotrophs like E. coli that can just quickly ramp up transcription and translation and grow fast. And substrate affinity, which means like they can take up substrates even if they're in low resource concentrations and grow slowly, efficiently, but slowly. And that was what we would call the oligotrophs. And the other thing I want to emphasize is I'm presenting this as 2 categories, but it's not 2 categories. There's a spectrum here. So we have sort of oligotrophs on the left, copiotrophs on the right, and we say, okay, let's say we have this imaginary y-axis that's selection for high rates of maximal growth. Again, the copiotrophs are built to grow quickly. When conditions are right and resources available, they can ramp up transcription and translation and grow fast. Versus oligotrophs, again, not invited to the feast. They can't take advantage of those conditions. They grow efficiently, not rapidly. Okay, the other caveat I want to mention is when I made this claim, most soil bacteria are oligotrophs, I'm mainly talking about bulk soil. So we're not talking about soil right next to roots. Plants are good carbon pumps, right? They're pumping all this carbon roots. So if you're looking at bacteria right in the rhizosphere or right in the root zone, you're going to get some of these faster growing, these copiotrophic organisms thriving. Likewise, if you look at the the area right around earthworms, right? There's going to be lots of carbon availability. We're not going to see these, or not see as many of these oligotrophs. Same, if we're talking about the, excuse me, this soil litter interface, where there's going to be elevated carbon availability and just generally elevated resource availability. But in general, I think in bulk soil, we're seeing a lot of oligotrophs. So what's the evidence for this? Okay, if you look at biomass in soil, it's impressive, right? So 400 to 2,000 kilograms of soil microbial biomass per hectare. This includes not just bacteria, also fungi and all sorts of other things. Number of bacterial cells, 10 to the 9th to 10 to the 12th bacterial cells per gram soil. So from this perspective, you look at these numbers and you'll be like, soil is just this dense rainforest packed with bacteria. Right? There must be a lot of resources to sustain those levels of biomass and that amount of bacterial growth. But let's look at these same numbers from a different perspective. Microbes occupy only, it's estimated they occupy only 10 to the minus 6% of available soil surface area. So from a if we shrink down to the size of a microbe and imagine sort of a soil landscape, it looks much more like a desert, right? There's some biomass here and there, but there's also a lot of barren patches. So this would suggest that sort of resource availability, resources are the norm in soils, not the exception. Other lines of anecdotal evidence. Most soil bacteria are small, less than half a micron diameter. Here's an example of they call it dwarf, we don't call that anymore, but small bacteria. Versus E. coli, this porker of a cell is living in the lap of resource luxury. It's big, and this is a well-established phenotypic response to resource limitation. Cells, bacterial cells in particular, become small. They're trying to maximize surface area to volume ratio. So again, this is just anecdotal evidence that a lot of bacteria in soil are resource limited. Other evidence. Most soil bacteria appear to persist in a dormant state most of the time. So this is sort of some estimates. If we look at the human gut, there aren't that many dormant bacteria. It's relatively high resource availability, flow through system, you have to grow fast. You can't sort of really be dormant and persist for very long in the gut. Versus in soil, about 80% of the bacteria are dormant. I don't want to get into what does dormant mean. Dormant just means less active. Doesn't mean they're dead. It doesn't necessarily mean they're in a spore state. They're just active, about less active. Again, evidence that resource limitation is the norm, and growth rates are probably pretty slow. But I asked this question, now I'm going to try to answer it. Okay, what is the average generation time of bacteria? So I posed this question to Tristan Caro, who was a graduate student that worked with me, did a rotation in my lab. I'm like, no one knows the answer to this question. Nobody knows even within an order of magnitude. Let's see if you can figure this out. So he was an expert in stable isotope lipidomics. Don't want to go too much into the details of the method, but essentially you add deuterated water, so deuterium instead of normal hydrogen isotope, and you add that to soil, you measure the incorporation of the deuterium into membrane lipids, and you can use that to estimate generation time in soil. So not in the lab, not sort of culturing it on a Petri plate, but in actual soil environment. A few details. So the reason this method works, well, first of all, it's been validated a lot with bacterial cultures. It's even been validated with people that have cystic fibrosis and looking at bacterial growth rates in sputum. But as far as I know, this is the first time this method's been used in the soil system. So it works because all organisms incorporate hydrogen from water into the alkyl chains of lipids. And we can use this isotope ratio mass spectrometry to quantify deuterium incorporation in less than 1/1000th of the generation time of the bacteria. So this is important because you can use a relatively short incubation time and still estimate generation times, even if the generation times are far longer. And third, lipids are unlikely to be resynthesized for maintenance and repair in the absence of growth. We know this from lots of studies. And the other thing I'll mention is we're just looking at intact polar lipids here. Lipids degrade really quickly when a cell dies, so we're just looking at alive, intact bacteria. And also for the purposes here, we're ignoring fungi. We're just looking at bacteria. But like all methods, there's caveats. So one caveat is the taxonomic specificity of lipids is limited. So I can tell you average generation time in a soil. I can't tell you, oh, this group of bacteria is growing a lot faster than this other one. And also we're adding this deuterated water to soil slurries. So we're likely accelerating microbial growth. So we're probably giving sort of on the short side of estimate of generation times, and real soil are probably growing slower because here we're sort of mixing the soil, lots of water availability and so forth. Okay, so to do this, I will say this method is very low throughput. It's hard to do a lot of samples here. So we picked 3 soils for this experiment: conifer forest soil, grassland soil, alpine tundra soil. And I'm not going to pretend that these 3 soils represent the full breadth of soils on the globe. These were soils that were relatively close to Boulder that we could collect fresh. And we know they're different. They're quite different in terms of bacterial community composition, soil properties, and so forth. Are you ready for the drum roll? Okay. So what's the answer? So first off, I will say in any given community, there's a broad range of estimated generation times. That's to be expected. You can ignore this. This is just showing deuterium incorporation to lipids over time. These are different lipid fractions, but we can calculate a weighted mean generation time for each of these soils. So for the conifer forest soil, 20-day generation time is what we estimate. 28-day for the grassland soil, and 90-day generation time for an alpine tundra soil. These things are not growing fast on average. Again, there's likely some bacteria that are growing fast, but on average, these communities are very slow to turn over. So John, I think you win the award. You're the only one that said long time. So again, I'll show this, and the take-home message is they're growing very slowly. 20 to 90-day generation time, depending on the soil. What we'd like to do is repeat this for a bunch of additional soils, but again, the method is fairly low throughput, so we're working on it, but it'll take a while to get there. Okay, so what are some of the implications for this? So again, I just want to emphasize that bacteria in soil, they're not like E. coli growing fast. They're really growing much slower. And so what are some of the implications of this? One is, and this will not be news to any ecologist in the audience, which means all of you, and that is biomass is not equivalent to growth. So just because you have long generation times does not mean you don't have high biomass. And we see some evidence for this. So this is data on soil microbial biomass carbon across the globe. And look at what we see in like high latitude systems. There's lots of microbial biomass in those soils. But again, that doesn't mean they're growing fast. It just means that biomass is accumulating. So if we use like a plant metaphor, right? You can have a forest with high biomass, but slow turnover and low productivity. Or we can have like a productive prairie system where we have low standing biomass, fast turnover, and high productivity. So we see that same sort of thing if we're talking about soil bacterial communities. Biomass of bacteria is not equivalent to how fast those bacteria are growing in that system. It's kind of obvious, but it's worth mentioning. Another thing, so if we're seeing these long generation times on average in soil bacterial communities, we wouldn't expect a lot of temporal turnover. If you go back to the same site and sample over time, you'd expect not that many changes. And that's exactly what we see. So former postdoc in my lab, Paul Carini, who's now at University of Arizona, he's sampled a north-facing slope and a south-facing slope at a subalpine site near Boulder. And we picked a site because it's really seasonal, sampled over a 9-month period every 3 weeks. And what he was trying to look at is what's the temporal variation in the soil bacterial communities and fungal communities, but I'm not going to talk about that today. And to make a long story short, so these are 9 by 9 meter plots. And we had to sample the hell out of them because we needed to capture spatial variation in order to see what's just spatial variation, depending on what day you sample, versus what's actually temporal variation. And the answer is the spatial variation across sub-meter scale in soil bacterial community composition is 3 times larger than temporal variation. And again, this is like soils under snow versus hot, dry summer conditions. So there is some temporal variation, but it's very hard to quantify because there's not much of it. Again, we know there's some taxa that exhibit temporal variation, but on average, that community just isn't changing that much over time. So some other implications. So in soil, like in many environments, most microbes are very difficult to study in vitro, like on a Petri plate. So I would argue one reason for this is that they're just not adapted to grow quickly on a Petri plate, right? So we can do, and just to emphasize the scale of this problem. So a former postdoc in my lab, Manu Delgado Bacarizo, he basically said, what are the most abundant soil bacteria worldwide? And he identified about 511 bacteria and archaea that are reasonably ubiquitous across the globe and very abundant in individual soils. And of these 511 bacteria and archaea, less than 10% of them have ever been cultured. And this is just showing genome match to cultivated isolates. And many of them are very distant from anything we've ever studied in the lab. So I would argue, again, one reason for this is just that it's really hard to grow them in the lab because they just grow slowly. Maybe it's because we're not mimicking the conditions for growth or the environment they need, but it could just be that they grow fine. It just takes a long time, right? So this is just a back of the envelope calculation of what it takes. So let's say you have that bacteria with a 10-day generation time, okay? And this is a model just based on sort of optimal growth conditions in the lab. We're assuming a visible culture is about a millimeter in size. We're assuming there's no death and there's no lag phase. So they're fully exponential. To see the time required for visible colony, so how long it takes before you can see something on a Petri plate to isolate it, we're talking about more than 200 days. Right? So it could just be there's lots of bacteria we could grow in the lab, just nobody has the time or patience to do this. So if you're a graduate student and your advisor says, I want you to try to grow this bacteria, and you think that it has a long generation time, run away. Because you don't want to be doing your PhD for 5 to 10 years, right? Before you can get enough colonies to actually do experiments with them. So I think it's fair to say that a lot of microbiology is built on this ability to grow microbes, including bacteria, in the laboratory and study them and look at their traits and environmental preferences. But a lot of these more oligotrophic bacteria, they just have long enough generation times. It's hard to get them to grow and study them in the lab. So the patron saint of microbial ecology, Don Rumsfeld, has this great quote: there are things we know. We also know there are known unknowns. As I say, we know there are some things we do not know. So I would argue a lot of soil bacteria are these known unknowns because we can't study them in the laboratory. Mainly, well, many reasons for this, but one reason could just be they just grow too slowly. They're not built for rapid growth. So how do you study bacteria that are difficult to grow in the laboratory? DNA sequencing, right? So we can use either genomic sequencing or metagenomic sequencing, extract DNA from a sample and try to piece together the genomes and figure out from the genomic data what that organism is doing in that environment and what sort of environmental preferences it might have. And so I'm going to give you an example of this, but I want to preface it with saying genomics and metagenomics are no panacea. I'm going to be the first person, and we rely on these methods all the time. But just because you have a genome does not mean you understand that organism and what it can do. It can allow you to generate hypotheses about that organism, but it's not the be-all and end-all, right? If I just give you a genome of a plant, can you figure out everything we know from years and years of studying that plant? No. Right? And the same is true for bacteria. But again, allows us to generate hypotheses. Okay, so let's try to find gradients of soil where we expect to go from a more copiotrophic dominated system to a more system more dominated by oligotrophic bacteria. So for this example, I'm going to talk about soil depth. So most carbon comes from plants. Most plants are at the surface or more surface horizons. So you have a lot more carbon availability at the surface than as you go down. So we go from high resource availability to low resource availability. Of course, many other factors also change with soil depth, but I think this is one of the more dramatic gradients. You don't have to go down that far. You can just go down a meter and you're getting to a system that has very limited resource availability. It's just not a lot of available carbon to fuel growth. So a few years back, we looked at two soil profiles down to a meter depth at each of these sites, broad range of ecosystem types, tropical rainforest to desert to agricultural sites, forests, temperate coniferous forests, and so forth. And we're looking to see how do these communities change with soil depth as we go from higher resource availability to lower resource availability. And one thing really jumped out. So this is a soil profile in South Carolina, beautiful soil profile, I will say. And you look with depth, so we have depth on the y-axis going from the soil surface to about almost a meter in depth. And one thing that jumped out is this is relative abundance on the x-axis. These deeper soils are dominated by a single phylum. It used to be called 83, now it's called Dormibacteriota. This is a phylum for which no cultivated representative exists today. It's an entire phylum of bacteria that's dominant in these deeper soils, of which we effectively know nothing about, because we've never been able to grow it in the laboratory and study its traits and so forth. And it turns out this soil in South Carolina is not unique. If we look at a lot of these other soil profiles, we see similar patterns, where the members of this group, this Dormibacteriota, tending to become the dominant group in the deeper soil depths. They're still found in the surface soils, but they're not that abundant. And the specific patterns differ, but in general, they become relatively abundant with soil depth. Okay, so I had a graduate student, Tess Brewer, who was like, Noah, that's great, but let's try to figure out what these bacteria are doing, these members of this phylum Dormibacteriota. Again, what she did was she extracted DNA from these samples. She did metagenomic sequencing, which is basically sequencing all the DNA, and tried to assemble the genomes just from members of this group. So this is like taking a library, sticking it in a blender, and trying to sort of reassemble an individual book in that library. It's challenging to do, but the reason it worked is because this organism is abundant, this group of organisms is abundant enough in soil that you can actually assemble genomes from it. And the first thing that she noted is that members of this phylum, again, we're talking about an entire phylum of bacteria here, is consistently more abundant in soils with low soil organic carbon. So this would hint that it is sort of a canonical oligotroph. It tends to become more abundant in soils with low organic carbon. And this is also true if you look at surface soils. So this is a collection of hundreds of surface soils from across Australia, where even if you just look at surface soils, members of this phylum tend to be more abundant or relatively more abundant in soils with low organic carbon. Again, just indication that it's a oligotroph. And she was able to recover the genome. And the first thing she saw is, first off, a lot of these genomes are novel. They're phylogenetically unique. But there is some similarity to some other genomes that have been assembled from other studies, including studies in thawing permafrost and in an Antarctic desert. Okay, so novel diversity, but again, I want to emphasize this whole phylum, we still do not have a single cultivated representative from this group. So all we know about this group is just inferred from biogeography, like I showed, or from what we think we can understand from the genome. So what do we think this bacteria can do? One thing is, as we would expect, it's predicted to have low maximum potential growth rates. So this is not growth rate in soil. This is what's its maximum potential growth rate if conditions were optimal. This is calculated by looking at codon usage bias. I won't go into those details now. And what she's doing is looking at the Dormibacteriota, which are these green ones here, compared to all the other bacteria that are common in soil. And it appears to have, it's not unique, but it appears to have low maximum potential growth rate. Even if you give it resources, it can't grow that quickly. Okay. It also has some unique features. It appears to have quite a few genes that code for carbon monoxide dehydrogenases. So in other words, it can take trace gases, in this case, carbon monoxide, and presumably, again, we're guessing this based on the genomes, we haven't actually been able to confirm this in the lab, use carbon monoxide, oxidized carbon monoxide, and use that as an alternate energy source. So I won't go into all the genomics and so forth, but I will summarize what we think we know about this group of bacteria from the genomes. They do appear to be aerobic heterotrophs, okay? They're, again, likely slow growers, and this could be why no one's grown it in the lab. It doesn't appear to have really unique substrate preferences. It's probably just grows really slowly. It has, again, these multiple carbon monoxide dehydrogenases that presumably allow it to use trace gases in the atmosphere, use that to sustain some low level of activity. It's capable of spore formation. Again, we're thinking it's capable of spore formation, which may allow it to survive periods of extremely low resource condition or other stressful conditions. And it has the ability, or at least has the genes for trehalose storage. This is a starch some bacteria can use to sort of store up resources when times are good and use that as an energy supply when resources become more limited. Okay. So this is one example of a group of bacteria that we think are sort of canonical oligotropes that are really abundant in soil. Where do we go if we want to find a whole soil community that's presumably oligotrophic? Antarctica. Okay, so I'm going to transition here. And unlike what you may guess from this picture, not all of Antarctica is covered in ice. So the regions shown here in the sort of the tan color show ice-free soils, right? And if you add it all up, Antarctica is a big continent, right? So even though it's a small fraction of the Antarctic continent, we're talking about more than 47,000 kilometers squared of ice-free soils. And these are soils that haven't been glaciated oftentimes for thousands and thousands of years. They've been ice-free and developing in place. And this is what these soils look like, okay? For scale, there's some people here, right? There's the closest plant is like 2,000 miles away, right? There's, as you can imagine, not a lot of carbon, right? Really stressful conditions for organisms to live. So a few years back, I had the opportunity to join a field campaign to Shackleton Glacier, named after the ill-fated explorers trying to get to the South Pole. And just for scale here, I want to emphasize that Antarctica is big. Right? This won't be news to anybody. So this is the major US base in Antarctica, McMurdo Base. This is where you have to fly in. This is a 9-hour flight, right? So Antarctica is huge, right? But we had the opportunity to go to this site, this is how I spent my summer vacation photos, right? This is the field camp. That was my little tent. We were there for about 6 weeks. And the reason we had to be there so long is the weather is so uneven. You can't just helicopter over to different places and so forth. This is the field camp. It doesn't exist anymore. It was all taken down at the end of this field campaign. Just an amazing landscape. So here's two people coming back from collecting the elusive soils there. Just spectacular landscape. What about these soils? This is the mountaineer that was keeping us from killing ourselves when we were out there. These are soils. So this is not just ground up bedrock, right? These soils have been developing in place often for thousands of years. As you can imagine, they have extremely low organic carbon concentrations. 95% of the soils we collected from this campaign had no measurable organic carbon. No measurable, right? There's probably some, but and they're often very salty. They have very high concentrations of nitrate salts, chlorate salts, and perchlorate salts. And these are salts that are just generated by atmospheric processes that deposit on the soil. And because they're so little liquid water, they just accumulate in that soil over hundreds and thousands of years. Most of the soils that are estimated to be more than 10,000 years old, which means they've been developing without ice on top of them for more than 10,000 years. And no liquid water. They do get snow. There are patches of liquid water, but most of that snow just sublimates. It's so dry. And that's why we know that, because there's so many of these highly soluble salts that accumulate in these soils. These systems just do not receive much, if any, liquid water. And just for scale, this is where we're sampling. We're sampling up and down this valley. It's essentially capturing an elevation gradient on all these different features, these ice-free features in the landscape. Again, just another photo because why not? I don't have any cute animals to show you, so you get cool landscapes to look at. But really dramatic landscapes. So at each one of these features, we would set up transects and sample, sort of mini elevation gradients within each of the features. And just for scale here, so these are the two sites here, sort of at the Ross Ice Shelf. So this is Antarctica, so everything's shifted. So South Pole's at the top, Ross Ice Shelf is down here. We ended up collecting about 204 soils collected from across the Shackleton Glacier region. And these are the two lowest elevation sites. This is what do you do with this? You have to flex it up, right? And I will say, I just want to say, this was a huge team effort. Some of you may know Marcy Adams and Byron Adams. They're at Brigham Young University. Some of you may also know Diana Wall, who unfortunately passed away 2 years ago, something like that. And this was her 34th consecutive field expedition to Antarctica. So it was really amazing. I got to spend 6 weeks with her in the field. And also I want to thank Nick Dragone, who did not get to go to the field, but he did all the work I'm going to talk about. Brought the samples back to the lab, and then he spent a few years studying them. Okay, take-home messages really quickly here. As long as we see a relatively strong elevational gradient in both the types of bacteria we see in these soils and fungi, which I'm not going to talk about today, and in the diversity of these communities. Diversity tends to go down as we go up in elevation across all these sites. Okay. And I don't think it's elevation per se. that's driving these changes in community composition and diversity. Instead, there's lots of things that co-vary with elevation. So as we go from the Ross Ice Shelf, relatively like basically sea level, up towards the South Pole, higher elevation sites, you see an increase in salt concentrations in these soils, increase in UV exposures, and a decrease in temperature. All these sites are cold, but the higher elevation sites are really cold. And also there is presumably some water available at the lower sites, but the upper sites, these are dry. They've probably been dry for thousands of years. So we then wanted to figure out, like, how are these microbes surviving in this system? Right? What traits must the bacterial communities have to survive in these conditions? So we picked 12 of the low elevation soils, relatively temperate by Antarctic standards, and 15 soils from these higher elevation sites. And we did all the metagenomic sequencing to try to say, okay, what can it tell us about what traits, what sort of community aggregated traits we're seeing shifting across this elevation gradient. And to make a long story short, we find that these higher elevation sites, we see a lot of genes for trace gas metabolism. So a lot of these taxa seem to have this ability to not just oxidize carbon monoxide, but also hydrogen gas, present in low concentrations in the atmosphere, but they can generate a substantial amount of energy from oxidizing these substrates. And this is a common feature that's been observed over and over again of bacteria that live in these really resource-limited oligotrophic conditions, where they've evolved the ability to oxidize carbon monoxide and hydrogen to survive and maintain some level of activity. Why hydrogen gas and carbon monoxide? These are ubiquitous substrates, very low concentrations in the atmosphere, but they're always in the atmosphere. They're good electron donors, right? You can get a good amount of energy from it, and they're highly diffusible. And that's important because they don't need specific transporters. These gases will just diffuse right into the intracellular spaces of the bacteria. Okay. So I mentioned that we collected 204 soils for this study. Only 162 yielded any bacterial and fungal DNA for analysis. So those results I showed you weren't from the full data set of 204. It was only 162 samples. And it turns out the sites at the higher elevation oftentimes dropped out. We didn't get anything. We didn't get any DNA that we could analyze and sequence and so forth. So my graduate student at the time, Nick Dragone, currently a postdoc working with me, when he first showed me these results, what do I say as a bad advisor? You did something wrong. Right? There must be some error on your part about this. And he like grumbles, makes that face. And so he goes back, he does everything he can, different DNA extraction, using different protocols and so forth. And the answer is the same. There's basically 20% of these soils, we couldn't get any DNA signal out of them, any microbial DNA signal. And again, it's not sort of random. It tends to be clustered at these higher elevation sites. So these are just pie charts showing the percent of samples collected from each of these features where there's no detectable biomass. Okay, so I was like, maybe there's something interesting here. So DNA sequencing is just one approach, and quantifying total amounts of DNA is just one approach. So he threw the kitchen sink at it. Sequencing, quantitative PCR, culturing, measuring ATP concentrations, adding labeled glucose and seeing if that glucose was converted to CO2, right, which is a very sensitive method for detecting any sort of microbial activity. And the results were all the same. About 20% of these samples had no detectable microbial biomass. I'm not saying they're sterile. Proving a negative is hard, right? There could be something in there. But using the best methods that we have available, there's no detectable microbial biomass in these samples. And so we're really sort of approaching the limits of life, as far as we can tell, or bacterial life. And it's, again, at these high elevation sites, so it's probably not just one factor. It's this combination of super, super dry, very low organic carbon, high salt concentrations, and extremely low temperatures. So together, they just make it, it's like a too tough a place for bacteria to live, or fungi for that matter. And this was surprising to us, because you look at a lot of environments that we would consider extreme, and there's bacteria there, right? Geothermal systems, deep subsurface systems, Atacama Desert. But here we have a case in Antarctica where we reach that point, that sort of limit of life, and we can't detect any microbes. Now briefly, so one of these photos is from the surface of Mars, one is from Antarctica, right? Which one is which? Which one's Antarctica? well, right, you can see a little helicopter over there. That's the tip off. But I want to emphasize this because there's a lot of interest in Antarctica as an analog for Mars. Like if we study Antarctic soil bacteria, it'll tell us something about what's living on Mars. I don't know, right? There are some similarities, but I don't want to sort of talk about that. I think there's two things to take home from this. One is if our methods, the same methods NASA is proposing to use on Mars, can't even detect bacteria in Antarctica, like will we be able to confirm with accuracy that, Martian surface is devoid of microbial life? I don't know. And the other thing I want to emphasize is you don't have to go to Mars to find crazy bacteria that are sort of working at this limits of life. You don't even have to go to Antarctica, right? Like a lot of these sort of general life history strategies you can find in sort of any soil. They tend to be more abundant in these systems like in Antarctica, but it's not unique to Antarctica. So some of these really oligotrophic conditions are more common in lots of soil environments. Okay, I know I only have a few minutes. So I want to briefly, this is new unpublished work that I think is pretty fun, but you'll be the guest of this, right? You'll be able to tell me if I'm right. So in the process of doing this work on Antarctic soil microbes, a question came up. Are there bacteria that are endemic to the Antarctic continent? We know there's lots of animals. So by endemic, I mean only found in Antarctica and not, as far as we know, detected anywhere else. So emperor penguin, endemic to Antarctica. This is the one cute picture you get of an animal, this entire talk. Lots of plants like this moss here is endemic to Antarctica, only found on the Antarctic continent. But are there bacteria that are also endemic to Antarctica? We don't know. Like presumably there's enough dispersal that this is not the case, that there aren't bacteria that are unique to Antarctica. But I think there's two reasons why we might find endemic bacteria in Antarctica. One is it's geographically really remote. Right, so the closest landmass to Antarctica is still more than 1,000 kilometers away, right? And this is just looking at the Antarctic Peninsula. And plus, the way that the atmospheric currents work, there's a lot of barriers to atmospheric dispersal onto the Antarctic continent. It's not easy for bacteria from Australia or Argentina to just get alofted and then get deposited on the Antarctic continent due to the circumpolar vortex. The other reason is this is not going to be unique to anybody, but Antarctica is really cold, right? So it's got these unique environmental conditions. Average temperatures on the Antarctic continent are like minus 50 C. So it's cold. Most of these sites that we've looked at, they never get above freezing, as far as we know. And also, of course, really dry. So some of these sites, on average, they receive about 15 centimeters of precipitation. Most of that sublimates before it ever enters the soil system, right? So it's not actually wetting the soil. So this combination of these geographic limitations and these unique environmental conditions may give rise to endemic bacteria. So we looked for one genus of bacteria, Arthropacter. We focused on this genus because it's widespread in soils across the globe. So this is a group of actinobacteria. It's also highly diverse. So if we look at just reference genomes that are already available from previous work, there's about 600 species level representatives that are out there already, and it's readily culturable. That's why there's so many reference genomes. Right? This thing is actually fairly easy to grow in the lab. So we're asking this question, are there bacteria of the members of this genus Arthropacter that are endemic to the Antarctic continent? So we looked at an expanded data set of soils from all across Antarctica that span a broad range of soil types and environmental conditions across the continent, about 120 Antarctic soils and 472 soils outside Antarctica. And we use metagenomic sequencing and we use this strain level detection. I won't get into the details to actually identify the specific species and strains of Arthropacter that are detected in each of these soils. I just want to emphasize, we have these 120 Antarctic soils. This is just showing the variation in mean precipitation and temperature between these Antarctic soil data set and the non-Antarctic soil data set. And we specifically included a lot of soils in the non-Antarctic data set that come from hot deserts, cold deserts, like the Tibetan Plateau, like Svalbard, Greenland, so forth, to say like, okay, then the question is, are there taxa that we can detect in Antarctica and we don't detect elsewhere? So out of all these metagenomes, we didn't detect Arthropacter in every soil. That's just probably just because they're too rare. But we did detect 143 different strains of Arthropacter across this huge collection of soils. So everything that's in yellow is a strain just detected in the non-Antarctic soils. Everything in blue is a strain that's just detected in Antarctica. And I know this just looks like some impressionist painting from a distance, but so let me summarize. Essentially what we found is 32 Antarctic strains that were only found in Antarctic soils. We never saw them outside of Antarctica. We found 170 strains that were only detected in non-Antarctic soils, and only 4 that were shared. And all 4 of these were found at a hot desert in Australia. Okay, so I don't know how, it's kind of tricky to do statistics on this, but basically there's very few strains that are shared between Antarctica and non-Antarctic soils. So this is evidence for at least this group of endemism in the group. We don't see these same species or strains of Arthropacter outside of Antarctica. They're really unique to the Antarctic continent. So the last question we're going to go through very briefly is like, okay, so if they are truly endemic, they might also have unique environmental preferences. So I already mentioned that this bacteria is relatively easy to grow. I say relatively because Mary Childress, who led this work, this was like months and months of work to get this strain collection of 30 Arthropacter, unique Arthropacter isolates from Antarctic soils and 30 non-Antarctic Arthropacter isolates collected from a wide range of other soils. And we focused on 2 environmental preferences, desiccation tolerance and temperature optima, because we thought if you're going to survive in Antarctica, you have to have high desiccation tolerance and you have to be adapted for growth at low temperatures, because Antarctica is cold. So first, desiccation tolerance. So this is, again, a lab study. We're going from wetter conditions to drier conditions. This is manipulated using polyethylene glycol and a Petri plate. Let's not worry about the detail. What I'm showing here is growth rate relative to the highest water availability, moistest conditions. And take-home message is the Antarctic soil bacteria are actually not more desiccation tolerant than the non-Antarctic isolates. In fact, sometimes it's the reverse pattern. So yeah, Antarctic Arthropacter are not more desiccation tolerant than non-Antarctic Arthropacter. So they all seem to be growing best at sort of higher water availability. It's not a huge differential between these Antarctic isolates and the non-Antarctic isolates. What about growth temperature? Antarctic isolates, as you'd expect, have much lower optimal growth temperature than members of the same genus from outside Antarctica. And you can see this, so temperature here is on the x-axis, and this is showing relative growth rate for each isolate. The non-Antarctic isolates tend to have a much higher temperature optima than the Antarctic isolates. And importantly, they also have lower maximum potential growth rates. The blue bars here showing Antarctic isolates are always lower. Even in the lab, they just don't grow that well compared to the non-Antarctic isolates. So to summarize, Antarctic Arthropacter temperature optima is much higher than average Antarctic temperatures. So I know in the back of the room you can't see the scale here, but I already said that the Antarctic isolates tend to have a lower temperature optima than the non-Antarctic isolates. We expected this. What we did not expect is their temperature optima is like 17 degrees C on average. These soils never see 17 degrees C. Again, they barely ever reach 2 degrees C. So what's going on here? I don't know. Short answer. But my expectation is that the Antarctic soil bacteria may only be active when conditions are really favorable. Maybe there's 1, 2, 3 days a year when there's enough sun exposure and the temperatures are high enough, there's like some elevated temperatures. That's when they're active, and the rest of the year, they're not doing much. And we do see that, so here's some liquid water on a really hot day. There is some of these sites where there is some snowmelt, some liquid water. And I think these bacteria, these arthropacteria, they're really just taking advantage of these short glimpses of time where they can actually grow. And that's why we see this mismatch between their temperature optima and the actual temperatures experienced in the system. So just to summarize, I think even for oligotrophic bacteria, timing is everything. And I think this is really important to consider when we think about climate change, which is occurring really rapidly in the system, and how that's going to change these communities. And it could have really dramatic changes, even if it's just a few more days that are warmer than is more commonly observed, at least historically, at these sites. So with that, I want to thank a lot of people that contribute to this work. I'm up here talking about really there's a huge group of people that contribute to the work that was presented today, and including lots of folks in my lab groups. They're an awesome group, so it's really been great to work with them. So thank you. I will say, if you're ever in Boulder, look me up. But hopefully we have some time for some questions. Jenny.
I was wondering if there were any
Yeah, I didn't talk about archaea because they're there, but really, really rare, and it's just a handful of soils. Fungi are a different story. So I kept the focus on bacteria just to try to keep it a little bit more concise. There are fungi, including at some of these higher elevation sites. We have a lot of isolates of them now, and they're pretty cool. Like there are all sorts of weird adaptations, but much more patchy in the distribution. But we do see them at some of the higher elevation sites, not the ones where we didn't see anything, of course, but at some of the other sites. Sasha.
That was awesome. Thank you. I was really interested in your result of seeing much more spatial variability than temporal variability in the microbe. I think that's biomass, maybe the biomass.
Community composition. Yeah.
And so thinking about functions of microbes that we do see changing on relatively short time scales, what do you think? What insight does that pattern, the lack of change, blend into what controls those, like soil respiration or something?
Yeah, so we were looking at community composition, the amounts and types of bacteria and fungi in those soils. I wouldn't say there's no temporal variation, but it's really hard to detect. It's super subtle, especially compared to the spatial. So one problem with studying soil is it's destructive sampling. We can't go back and sample the exact same site every week or month, because by definition, you've taken that soil and removed it. Ocean, you can do that, right? So in terms of community composition, there's not much temporal variation. Activity for sure, right? If you measure CO2 at these sites, right, it's much higher in the spring than in the middle of winter or in the hot, dry summer, right, for sure. But in terms of like the community membership, there doesn't seem to be. And I will say also, I didn't mention it, but they also removed any extracellular DNA. So we're just looking at DNA from intact cells. But the activity for sure probably fluctuates among community membership, but just the overall structure of the community doesn't seem to vary that much with time, which is good news if you're studying, because you don't need to study it. If you want to characterize what soils in a given forest, you probably don't need to go back every week and sample. You can probably be pretty confident that, yeah, there's some temporal variability, but any spatial variability is probably gonna be a lot larger. Right. And I think it's just related to these things aren't growing that fast. Or the bulk of the community isn't changing that quickly or growing that quickly. Peter.
So there's this whole thing I've heard say about bacteria, everything's everywhere all the time. It's like, no.
I think that was presented as, what is that, just to be antagonistic, right? So anyone that's ever been sick, you know that's not true, right? Pathogens aren't just like everywhere all the time, right? So, you know, like that's the entire field of epidemiology is it's like there are these sort of spatial and temporal patterns and pathogen presence, right? And that's definitely true for organisms. I think sometimes we don't see that because, you know, for example, if you just focus on species level, which is pretty broad, yeah, you see oftentimes the same species everywhere. But once you dive down to the strain level, we see that that's not the case. So yeah, there probably is a lot of dispersal of microorganisms that can occur, particularly if they're capable of forming spores. But like every other organism on the planet, they're not just everywhere, right? I don't know if that really answered the question, but I mean, I think the Antarctica is one in a long line of evidence to show that that's probably the case, even at the continental scale. But there's taxa that seem to be restricted just to Antarctica. And as far as we know, we can't detect in other soils from outside Antarctica. Yeah.
Yeah, your number of dormant microbes. Is that across is that not all groups are dormant at the same time, or is it particular groups are more likely to be dormant than others?
Okay. That's a great question. I wish I knew the answer. So first of all, I think dormancy we have to consider that with a large grain of salt, right? Because dormant doesn't mean not active. It just means over the time course that we did a study, we didn't measure active growth. So I think by that same definition, a redwood tree would probably be considered dormant. Yeah, there's some growth, but not a lot of growth, right? It's probably a strange metaphor, but I think you get, so it doesn't mean that like all those things are not doing anything. They have to be maintaining some level of activity just to like stay alive and maintenance and repair and so forth. I think, and I know Jay Lennon has done a lot of work on dormancy, he would agree with that. But for sure, there's probably fluctuations in when an organism is less active or dormant and when it's more active. And I think we see some of that evidence for that in Antarctic bacteria, where they're probably just taking advantage of these sort of glimpses in time when the conditions are more favorable for growth. And I think that's true not just in Antarctica, but in a lot of soils. They're kind of their activity, but it's really hard to measure activity at the cell level in the field. But that's a great question. Yeah. Yeah.
So your lab data set that you're showing for temperature optimum being greater than the average, substantially greater than the average temperature of the environment. Was that temperature optimum for growth or for activity?
Or growth.
Growth.
Right. Yeah, it's for growth because we're looking at basic changing colony size, pretty rudimentary metric of growth. We've also used, I didn't show it, but for all of these isolates that we have from Antarctica, we've also used genome-based metric of temperature optima. It's a little tenuous. Also, those results are similar. It's still much higher. But when we apply this to marine systems from Antarctica, their temperature optima for growth is 2 degrees C. So it's something about that soil environment, like I mean, this is a cold environment. It's always cold, but for whatever reason, and maybe it's just the temporal variation where they're able to grow when it's warmer. Maybe it's other constraints on growth besides temperature. I don't know, but it's, as far as we can tell, and other groups have looked at this, there aren't true psychrophiles in Antarctic soils. So that tells us something about climate change responses. If temperatures go up a few degrees C, these things are going to be happy as clams. They're not true, as far as we know and can tell, they're not true psychrophiles. Yeah, way in the back.
But you were describing like the actual coverage of microbes in the soil, like not very much, it's like all like a desert. In that case, why is it surprising that not all of the Antarctic soils have microbes? You know what I mean?
Yeah, were you reviewer number 2 on our paper? Right, so it's a completely reasonable point. So I tried to be careful to say, I'm not saying these soils are sterile. I'm just saying they're below the levels of detection using this wide range of methods that we know work in really low biomass systems or have been proven to work. Because someone could argue, well, what if you took a kilogram of soil, extracted all the DNA, you'd probably detect at least a spore in there, right? And that's true, right? So I'm tried to be careful to say no detectable biomass, but you're right. Like I mean, microscopy is really hard, particularly in these sorts of systems. But yeah, it's true. If you spent weeks scanning around, you probably could find some cells in there, but it's just below the limits of our ability to detect living microorganisms in that system.
I think I was asking like right at the beginning of the presentation, you said there was like
Oh yeah,
this, they cover less of the surface than you would think.
right.
Then how come, wouldn't you expect like a lot of your samples?
Oh, well, sorry, what I was trying to say is like there's a lot of biomass per gram soil, for example, in terms of numbers of cells and amount of carbon that's in microbial biomass, but there's enormous amount of surface area in soil, right? Just because it's, so when you calculate it, and also bacteria are small, right? So you look at it from a surface area perspective, it's relatively low, even though biomass levels per gram soil are high. Yeah.
I think that's all the time.
All right, thank you.